Bismillahirrahmanirrahim. Today our lecture is about genotype environment interaction or gene environment interaction. So let's start our lecture. Before going into the detail of gene environment interaction, we should know what is genotype and what is environment. As we studied this in our previous classes, classical genetics, about the genotype, about the phenotype, different types of genotype, different types of phenotypes. So you know well about the genotype and phenotype, the basics of genetics. So let's remind uh, those things. Genotype is basically the genetic makeup of an individual. All the alleles possessed by an individual constitute its genotype. So the genetic makeup of an individual is basically its genotype, or we can also define it as any combination of two alleles. Any combination of two alleles of different forms of a gene at a locus in an individual is called as genotype of that individual for that locus. So it is basically the genes or genetic expression of an individual, or any combination of two alleles of different forms of a gene at a locus in an individual is its genotype. Genotypes, as we study already, that it can be heterozygous or it can be homozygous. When the both alleles are dominant or both alleles are recessive, then this will be homozygous. And in case of heterozygous, when one allele is recessive and one allele is dominant, then we can call it as heterozygous. Then the next is environment. Environment is what is around something. This can be living or this can be non-living. It includes physical, chemical or other natural forces. And these natural forces constantly interact with the living. Now, so anything that is around us is our environment. These environments can be living, or these factors can be living or can be non-living. These include the physical factors, the chemical factors, or other natural factors which affect an individual or which affect an organism is the environment. And these factors or these environmental forces, they constantly interact with the living organisms. Okay, basically there are two types of environment. One is macro environment and the other one is micro environment and the other one is macro environment. We will study in detail about micro and macro environment. Micro environment is basically the direct environment or the immediate factors which affect an organism within an ecosystem is its microenvironment. For example, a microhabitat, a very small specific area in a habitat, is its microenvironment. The immediate environment of any individual is its microenvironment. It is distinguished from its immediate surrounding by factors such as amount of incident light, degree of humidity, and the ranges of temperatures. Okay, so this is its microenvironment. And in microenvironment, as we said, that it is the immediate or direct environment which affect an individual is its microenvironment. So it differs from one individual within a population, from one individual to another individual. The environmental, the microenvironment of mine can be different from yours. Even within your hostel, for example, within your hostel, the environment of one room can be different from the environment of the other room. Okay, so you may now understand what is microenvironment. So the immediate or the direct environment of any individual within an ecosystem is its microenvironment. While on the other hand, the macro environment is environment associated with variables having large or easily recognizable effect is termed as microenvironment. Or we can also define the macro environment as larger or more general factors that affect the entire ecosystem. Any environment that affects the entire ecosystem, the entire environment, for example, the micro environment within a hostel from one room to another room is different. Okay, the factors which influence that a student, it can be different from one room to another room. Okay, now in case of macro environment, the environment of the whole university, of the whole ecosystem, those factors which are larger uh, are more general factors which affect the entire ecosystem or the entire university. For example, nowadays what we are doing online classes. It is due to what? It is due to some sort of changes in the whole political system, or other in some sort of changes in the whole Pakistan, which is affecting our study. So that is macro environment. Larger or more general factor that affect the entire ecosystem is called as macro environment. It includes differences over period, differences over season, differences over locations. Okay, so this is our macro environment. Now coming to the main topic, gene environment interaction or genotype environment interaction. It is basically when two different genotypes respond to environmental variation in different ways. Genes plus environment, it gives us its phenotype. We will study this in detail in next section in the preceding slides. So genotype environment interaction, the basic definition for your understanding is when two different genotypes respond to environmental variation in different ways. What are those ways? It is the expression of that genes within different environment. It will produce its phenotype, and its expression will be its phenotype. It can produce, for example, in case of maize, it will have high production in one environment. If you sow that gene or that the similar gene or the similar seed in different environment, it will give another response. For example, low yield. So in different environment, when two different genotypes respond differently to environmental variation in different ways, it is the interaction between gene and environment, genotype and environment interaction. Two different genotypes in one environment, it will respond differently. Or one gene in two different environments, it will respond differently. For example, in one it may have high yield, in one environment it may have low yield. So this interaction is gene environment interaction. The model for genetic model for quantitative traits, P is our phenotype. P is equal to G plus E plus G into E. P is phenotype, G is genotype, and E is environment. So we can say that phenotype is controlled by or governed by genotype, environment, and genotype environment interaction. Okay, so on the basis of model, the genetic model, we can define genotype environment interaction as phenotype is controlled by or governed by genotype, environment, and genotype environment interaction. Or we can say that phenotype is function of genotype and environment. Okay, what is phenotype? Phenotype is function of genotype and environment. Okay, we can say that phenotype is function of genotype and environment. Genotype, we studied already the genetic makeup of an individual, of an organism. Phenotype, the expression of that genotype of the gene, or that genotype is its phenotype. What is environment or environmental variation? Changes in the environment. It can be physical change, it can be chemical change, biological change, behavioral pattern, or life events. This is a detailed. So changes in environmental variation or changes in the environment. So P is equal to G plus E. Over here you can see the example: genes, environment, and the interaction of these genes and environment, it affect an individual, or it affect the phenotype. So we can also say that, or we already studied in the definition, that genotype, two different genotypes respond differently in different environmental conditions or in different environments. Over here in this graph we have three different environments: E1, E2, and E3. These are three different environments. These are three different environments, and we have genotype 1 and genotype 2. You can see over here in environment 1, genotype 1 has higher phenotype or high yield. For example, over here our phenotype is yield. Our phenotype is yield. Okay, so genotype 1 in environment 1 has higher yield, and it drastically reduced in environment 2. Okay, in environment 2 it drastically reduced, while showing the same genotype in environment 3, it again produces higher yield. While for genotype 2 we have high yield in environment 1, similarly yield in environment 2, and slightly reduced yield in environment 3. So we can say that for genotype 2, in every environment, in all three environments, there is no change or no difference or no or slight change in each environment for genotype 2. But for genotype 1, we can see that there is a huge difference in environment 2. There is difference in environment 2. So this means that genotype 1, it responds differently in environment 2. Okay, it responds differently in environment 2 for genotype 1. So this is basically for genotype 1, there is environmental interaction. So in environment 2 we can say that there is genotype environment interaction for genotype 1. A more precise definition of genotype environment interaction, we can say that it is dependent relationship between genotype and environment in which the difference in performance between two genotypes changes from environment to environment. Keep this thing in your mind that the difference in performance between two genotypes changes from environment to environment. You can see this in our previous slide. Okay. So the difference in performance between two genotypes changes from environment to environment. Maybe you don't understand this over here, but in the preceding slides we will elaborate this definition that the difference in performance, the difference in phenotype between two genotypes changes from environment to environment. Two different genotypes respond differently to environmental variations. Okay. The same definition as we studied earlier, that in two different environments, it responds differently. Okay. Next slide. We will discuss two situations, situation 1 and situation 2. For example, suppose you have two different genotypes, G1 and G2. Okay, you have two different genotypes, genotype 1 and genotype 2. The yield of genotype, the average yield or the mean yield of the genotype 1 is 500 kg. For example, the yield of a wheat in is 500 kg, while the yield of wheat of a crop is 1000 kg, of G2 is 1000 kg. The yield of G1, genotype 1 is 500, or crop is 500 kg, and G2 is 1000 kg. When it is sown in environment 1, its yield increases to 600 plus 100, its yield increases plus with 100 kg. Its yield increases 100 kg. So the performance or phenotype 1 for G1 in environment 1 is 600. Okay. In environment 2, suppose it is reduced by 100 kg, minus 100, and the overall performance of G1 in environment 2 is 500 minus 100 is 400 kg. The difference in P1 and P2 in two different environments is 200. Okay, the difference is 200. Now let's come to the G2. Its yield is 1000 kg. In environment 1, its yield increased by 100 kg, similar to environment 1 in G1, and its yield is reduced in environment 2 by minus 100. Okay, its yield is reduced by minus 100, and the overall performance for P1 in environment 1 for G2, as it is increased by 100, it is 1100, while in P2 it is 900. Okay, while it is 900. and the difference is 200 kg. The difference is 200 kg. Now by the definition of G and E, it is the difference of two genotypes. It is the difference of two genotypes with environmental variation. With environmental variation, the difference over here is 200 kg. The difference over here is also 200 kg. This means there is no difference for genotype 1 and genotype 2 in two different environments. The difference over here is 200. The difference over here is also 200. This means that there is no difference, so we can say that there is no gene environment interaction for G1 and G2. For genotype 1 and genotype 2, when there will be difference, then we can say that there is gene environment interaction. But over here, both the differences are same. This is 200 kg, this is 200 kg. Both the differences are same. So this means that there is no difference. So we can say that there is no gene environment interaction. Now this will be clear more. Let's go to the situation 2. In situation 2, we have the same G1 with 500 kg, G2 with 1000 kg, G2 with 1000 kg. In environment 1, its yield increases to plus 100, and in environment 2, its yield is reduced by minus 100. Suppose we have gene environment interaction for G1 E1 as plus 50. G1 E1, we have plus 50 gene environment interaction. G1 E1 is plus 50, while G1 E2 is minus 50 kg. Suppose we have this gene environment interaction, then P1 will be 500 plus 100 plus 50, 600 plus 50, 650. Okay, this is our phenotype 1 or performance 1 for G1 in environment 1. G1 in environment 2, as we said that the yield is reduced by 100. G1 E2 interaction is minus 50, so it will be 500 minus 100 minus 50. So this will become the performance 2 or P2 will be will be 350, and the difference between 650 and 350 is, the difference is 300. Now come to G2. In G2, we have 1000 kg of the 1000 kg of G2. The yield of G2 is 1000 kg. The mean yield is 1000 kg. In environment 1, as we said earlier, it is plus 100. The yield is increased by plus 100 kg, and environment 2, the yield is reduced by minus 100 kg. And G2 E1, suppose we have plus 200. This is based on calculation. If you want in detail, then I will give you the details. I will give you the detailed notes on these statistical calculations. But for here, now suppose that we have plus 200 kg of G2 E1 interaction, and G2 E2 interaction is minus 200 kg. Okay, G2 E2 is minus 200 kg. According to the genetic model, G2 plus E1 plus G2 E1, it will become 1300 kg. The P1 or the performance 1 or the phenotype 1 is 1300 kg. G2E2 is minus 200. So according to the genetic model, G2 plus E2 plus G2E2. So it will be 1000 minus 100, 900, and minus 200, 700. So our P2, the performance 2 or phenotype 2 is 700. 1300 minus 700, the difference is 600. The difference is 600. So now we can see that the difference between two environments, there is clear difference between the two environments for the phenotype, for the yield, 300 and 600. So the difference between phenotypes of two different genotypes in different environments is G into E. So we can say that in situation 2, the genotype environment interaction or gene environment interaction exists. Okay, now hopefully you understand the genotype environment interaction. Go to the definition and then see these examples, situation 1 and situation 2. You will see the clear cut difference in situation 2 while there is no difference in situation 1. Now why we study the genotype environment interaction? Now hopefully you understand the genotype environment interaction. Now why we study the genotype environment interaction? The purpose of genotype environment interaction. First of all, to investigate the relationship between genotype and different environmental changes. Okay, so you saw the gene in environment 1, environment 2, and you see the difference for two different genotypes in both the environments. So we investigated the relationship of genotype, how it will respond in different, how it will respond in terms of yield in different environmental changes or in different environment. Now to study the general adaptability. To study the general adaptability. This means to identify the genotypes which perform well over a wide range of environments. What do you mean by wide range of environments? For example, if we want to produce a wheat crop which should yield, its purpose is to develop a wheat crop which should give good yield globally for global production in wide range of environments, for global production in wide range of environments. So the purpose is to identify the genotype, to study the genotype, to identify the genotype which perform well in a wide range of environments, to study the general adaptability of a crop, of a breed. Okay, so to study the general adaptability and to study the specific adaptability. From specific adaptability, we means that in a particular environment, in Hazara region, in Peshawar region, in Lahore region, in Sindh region, in Karachi, so in a particular environment, in Sri Lanka, in Afghanistan. So in a particular environment, we identify the genotype which perform well in a particular environment. So we study the specific adaptability of a genotype, okay, of two genotypes, of different genotypes. So the purpose is to investigate the relationship of genotype and environmental variation or environmental changes. The second is to study the general adaptability of a gene, of a genotype or to identify the genotype which perform well in wide range of environments, and to study the specific adaptability of a genotype, or to identify the genotype sorry, that perform well in a certain environment, in a particular environment. Okay, hopefully you understand the types of the study, the purpose of gene environment interaction. Now let's go to the next slide. So now we will study the types of genotype environment interaction. Okay, for example, we have two environments, E1 and E2. Its performance or its response of two genotype, phenotype. Okay, it on the y-axis it is its phenotype or it performance, and we have two genes, G1, gene A, and genotype B, gene B. When the performance of gene A and gene B in two different environments is parallel to each other, okay, when it is parallel to each other, then we can say that there is no interaction. Okay, so the type 1 is type 1, you can say it as type 1, there is no interaction. When the performance of gene A and gene B is parallel to each other in two different environments, then we can say that there is no interaction, or it is type 1 interaction. Okay, no interaction or type 1 interaction. When there is interaction but no rank change, environment 1, environment 2, environment 1 and environment 2. In environment 1, for example, we have this 1, 2, 3 and 4. In environment 1, yield is, for example, 2 ton, 3.8 ton environment 1, its yield increase to 4.2 in environment 2, almost to 3.6, for example, 3.6 tons increased in environment 2. So we can say that there is no crossover. Both the gene, the performance of both the gene are responses. In responses, there is no crossover, but there is difference between the two responses in environment 1 and in environment 2 for both the genotypes. So we can say that there is interaction in performance for both the genes. There is difference in performance for both the genotypes in two environments, but there is no crossover. We call it as type 2 interaction. Type 2 interaction, when there is interaction but there is no crossover and there is no rank change. From rank change, we can say that if the gene B has changed from here for example, to this place, then we can say that it crossed the genotype A, it, its rank is changed, or this A is the performance of A is reduced over here. We can say that there is change in the rank for A and B. B is over here and A is over here. The A change its position to a lower position than B. We can say that there is rank change. Okay, so there is no rank change for A and B, but there is difference in performance. There is interaction. There is difference in performance. Over here we have no difference in performance. Keep in an eye on the example which we studied in situation 1. There was the difference was same. The difference over here is same. These are parallel to each other. The difference is, when these are parallel to each other, the difference is same. Okay, the difference is same. So we say that there is no interaction. But over here the difference is the difference is there. So we can say that there is no rank change, there is interaction, and there is no crossover. We can say that it is type 2 interaction or non-crossover interaction. Okay, type 2 or non-crossover interaction. This is parallel interaction. No interaction. This is non-crossover interaction. Now the type 3, the crossover interaction. The type type 3 or crossover interaction. There is rank change. When there is rank change, there would be interaction. Okay, E1 and E2. E1 and E2. You can see that the performance of A is increased from E1 environment to E2 environment. The performance of B genotype is increased from E1 environment to E2 environment. The performance is increased in E1 in E2 as compared to E1. There is high yield for A genotype in E2 environment as compared to E1 environment, and we can see that there is a crossover. The rank over here, for example, this is 1, this is 2, this is 3, and this is 4. Let's see this. These are ranks. From 1, the rank of B is changed from 1 to 4. And 2 to 3. 2 to 3. So 1 to 4. There is rank change. The rank of A is 3. The rank of B is now 4. So there is rank change. Okay, there is rank change for B, which was 1, now it is 4, and A is still in the same rank, 2 to 3. There is no change. So we can say that type 3 interaction or crossover interaction is there is rank change, and there is interaction. So let's summarize this thing. We have different types of gene and environment interaction. Type 1, when the genes are parallel to each other, there is no interaction. Type 2, we can, or non-crossover interaction, when there is no rank change but there is interaction. And type 3, there is rank change and there is interaction. Type 3 or crossover interaction. Okay, when there is crossover, then this is type 3 interaction. Okay, now a little bit about now a little bit about the history. Mather and Jones in 1958 have given the description of gene-environment interaction. This can be specified with simple case of two genotypes, G1 and G2, both raised in two environments, E1 and E2. Let's see this in detail. Number first is linear relationship. When the phenotypic difference between two genotypes G1 and G2 in two environments remain unchanged, the rank order of the genotype does not change. You can see in these two examples 1 unit decrease and 2 unit decrease, 1 to 2 and 1 to 3 for genotype 1, G1, G2. Similarly over here, G1 and G2. Okay, so both are almost parallel to each other. So it is linear relationship. When phenotypic differences between genotypes in two different environments remain unchanged, when these are parallel to each other, this is linear relationship. Non-linear relationship, when the change in phenotypic values of two genotypes in changed environment or in two different environments is in opposite direction or in the same direction but are unequal. Okay, when there is two different types of relationship, linear relationship and non-linear relationship. In non-linear relationship, when the phenotypic values of two genotypes in change environment is opposite or in same direction but these are unequal, we call it as non-linear relationship. We will study this in detail with example. In first example, when the change is in the same direction but unequal, this lead to a change in rank order. For example, G1 and G2, G1, G2 in E1 and E2 environment. The change is in the same direction. The direction of the change is same. Both are reducing or both are increasing. This can be both. The change will be in the same direction. Both are decreasing or both are increasing. Okay. Over here both are decreasing. This is 4, 1, 2, 3, 4. Both are decreasing. So G1 from rank 1 it goes up to rank 4, G2 from rank 2 to rank 3. So first type of non-linear relationship is change in the rank order when the changes are in the same direction. Okay, and the changes are unequal to each other. 1 to 4 and 2 to 3. So this is first nonlinear example of first nonlinear relationship. When the change is in opposite direction but does not change the rank order. Both the changes, this one is from 1 to 2, this is 4 to 3. One is increasing from 1 to 2, or we can say from 2 to 1, decreasing. One is increasing and the other one is decreasing. Both are in opposite direction. One is increasing, the other one is decreasing. Both are in opposite direction, but being in the opposite direction, but it is not changing the rank order in two different environments. If we have environment 3, there would be possibility that if it is changing in the same, there would be possibility of the crossover. There would be possibility of change in the rank order. This would become 4, this would become 2 or 1. So there would be possibility. But over here now there is, we are studying up to environment 2, so there is no change in the rank order. No rank order is changed. So when the change is in the second type of nonlinear relationship is when the change is in opposite direction, but there is no rank order change. Now the third type, when the change is in opposite direction and there is, which lead to the rank order change, as we studied previously, from 1 to 3 and 3 to 2 in environment 1 and environment 2. Okay, G1 and G2. The rank order is 1, the rank order over here is 3. So its rank order is changed from 1 to 3, while over here the rank order is changed from 3 to 2. Sorry. So over here the change is in opposite direction, but it is leading to the change in the rank order. Okay, so this is basically your gene environment interaction lecture. There are other details too based on the on these. You can study these for your understanding. You can study these slides. I'll share it with you. This is just for your understanding, but your lecture is up to 21 slide. Okay, you can study these interactions, gene environment interaction, the detail, methods to analyze gene environment interaction, genetical, traditional, genetic designs, adaptive study, twin studies, family studies, adaptation, when an individual is adapted in two different environments, twin studies, when two twins are given two different environments, when the environmental effects on children. These are the examples. So you can study in detail. I'll give you these slides. And molecular analysis, interaction with single gene, interaction with multiple gene, and genome-wide association studies with genome-wide interaction studies. So the examples: gene environment interaction, an example of lack of gene environment interaction. You just go through these slides, but remember the first few slides. Thank you very much for your time and for your interaction. Hope you have enjoyed this lecture. Okay, we will now conclude the session over here. Thank you very much.